Hello, good day class. I welcome you to the class today. After looking at the structure and the function of carbohydrates and looking at the structure and function of starch and also looking at the structure and the function of proteins and fats. So today we want to look at how we can test a food sample if it contains starch, if it contains reducing sugar, if it contains protein, or if it contains lipid. You know, lipid is fat, contains fat. So there are different ways that which we can test for carbohydrate, protein, or fat in a food sample. That is what I am going to be explaining to you, how to do it and what to look out for. This section is a practical section and you are going to handle the practical very soon. So in the practical, you are still going to, it will be assumed that you know this and you are going to carry the test out and you are going to observe this food test. So we can determine the presence of carbohydrates, proteins and lipids in food with a variety of food analysis or food tests. Primary tests for foods are as follows. Number 1 is the Benedict test for reducing sugar. The B is a test for starch with iodine. Then the third test is the protein test using Biuret, the Biuret test. Then we have the lipid emulsion testing also that we are going to look at. Now, for any of these tests, there are certain things that you need to put in place. And number 1 thing is that you have to crush the food sample with a pestle and a mortar. Then you have to combine the mashed food with distilled water in a beaker, just a little water. Then you use a glass rod to stir the crushed food so that the crushed food is going to dissolve and mix with the water. Then some of the food you see dissolving. Then you have to use a funnel and filter paper to strain the solution to remove the solid bits of food left over. Some of these tests may not involve crushing. Your demonstrator, the practical technologies that will demonstrate to you, is going to explain all that to you. But also you can crush all the food as explained here for you to get the answer. The final solution is ready for use in the food test or food analysis. Number A, as I said, is a test for reducing sugar, and that is the Benedict test. If you need to detect reducing sugars, you can use Benedict's solution. Benedict's solution is a dark blue alkaline chemical reagent. Reducing sugars include monosaccharides and certain disaccharides. These are the tests for reducing sugar. Number 1, set up a Bunsen burner to heat water to at least 80 degrees C in a water bath. In a test tube, combine some of your sample with Benedict's solution. Leave the test tube for 5 minutes in a water bath. Observe and record the color changes that you see. If your sample does not contain any reducing sugar, the resulting solution will retain its original color. If it contains reducing sugars, depending on the concentration of reducing sugar also, it will either turn green, yellow, orange, or brick red. So that is how to test for reducing sugar in food. The next test is test for starch with iodine. The analysis of organic samples for the presence of starch or complex carbohydrates is as follows. The first thing you have to do is scoop out a small amount of your sample and place it in the test tube. Second, put in some iodine solution. The third is to make note of any deviation or changes in color of the sample. A dark blue or black color indicates the presence of starch in the sample. The next test is a test for protein, that's the Biuret test for protein. Peptide bonds which link amino acids are detected by the Biuret test. For the experiment, we'll be using sodium hydroxide and copper sulfate. You start by dissolving your sample of interest in a test tube with the same volume of sodium hydroxide. Second is to stir for a few minutes after adding 2 drops of copper sulfate solution. Then you have to observe any color changes. The presence of proteins in your food sample will cause the color of the solution to shift from blue to purple. If the test is negative for protein, the blue color will be retained. The next one is the emulsion test for lipids, or the test for fat. Water does not dissolve lipids. Lipids do not dissolve in water. However, organic solvents like ethanol and others, and many others, will help dissolve lipids, will help dissolve your fat. The first thing you should do is to dilute some of your test sample with ethanol and place it in a test tube. The next thing is to ensure proper mixing. Then, to ensure proper mixing, you have to shake the sample vigorously and not properly. The third is to pour in a similar amount of distilled water into the sample. Then you make a note of any findings in your result. You observe the changes that occur. If your sample contains lipids, you will see a white cloudy emulsion. There are no lipids in the sample if there is no cloudy white emulsion. So this is where we're going to stop on the test for protein, test for starch, test for reducing sugar, and then test for lipids. Very, very shortly, you are going to handle all these, and you are going to see them as I've explained them to you. So go over your materials, and we're going to see in the next class. Have a good day.